Good morning, my dear friends. Today we are going to understand biomolecules chemical analysis. Friends, this is my second lecture on biomolecules. So, as we already know that biomolecules are most important and essential organic molecules that are present in the living system. Biomolecules are involved in maintenance and metabolic process of the living organisms. For example, proteins, carbohydrates, lipids and nucleic acids. Now it's the time to understand the biochemistry of biomolecules. So let's get started. Friends, chemical analysis of biomolecule means it's a process of studying different compounds, whether it is organic and inorganic, in the biomolecules, their molecular formula and their structure. Friends, we know a cell, whether it is a bacterial cell, whether it is an animal cell, whether it is a plant cell, these are all composed of some organic molecules to perform various functions. Friends, an experiment was conducted in which a living tissue, whether a vegetable or a piece of liver is taken and it is grind it in trichloroacetic acid with the help of this mortar and pestle, and we obtained a thick slurry. Now, in this chemical analysis, what we have done, we have taken this thick slurry and we have filtered through the cheesecloth, and after filtration we obtained two fractions. First was retentate and second was filtrate. Retentate, it is also known as acid insoluble fraction, and the second one is known as filtrate. Filtrate is acid soluble pool. Friends, so after conducting this experiment, we obtained two fractions. First is acid soluble pool or filtrate. After chemical analysis, we found that this filtrate or acid soluble pool is basically composed of micromolecules. Micromolecule represents roughly the cytoplasmic composition. For example, amino acids, simple sugars and nucleotides. Now we come to acid insoluble pool or retentate or pallete. It is basically composed of macromolecules and it is composed of polysaccharides, proteins, nucleic acids and lipids. Basically, this is the fraction which is composed of cytoplasm plus cell organelles. So it's very important. Now, so these two fractions that we are getting after conducting this experiment, filtrate and retentate, this represents the entire composition of living tissue or organisms. After conducting this experiment, we came to conclude that biomolecules are composed of micromolecules and macromolecules. Micromolecules means small sized, low molecular weight, found in acid soluble pool. For example, amino acid, sugar, nucleotides, minerals, water and gases. These all are found in acid soluble pool. Apart from organic substances, we are also getting some inorganic compounds like phosphate, sulfate. We are also getting this in acid soluble pool. Now, what we are getting in acid insoluble pool, as the name suggests, acid insoluble pool. So big macromolecules we are getting in acid insoluble pool whose molecular weight is greater than 10,000 daltons. For example, carbohydrates, lipids, proteins and nucleic acids. So friends, one exception is there. Molecular weight of lipids do not exceed 800 dalton, but we are getting lipids in acid insoluble fraction. My question is why? After filtration, we are getting two fractions. First is acid insoluble pool and the second is acid soluble pool. But the molecular weight of lipids do not exceed 800 dalton, but we are getting these lipids in acid insoluble fraction. My question is why we are getting? The answer is, when we grind the tissue in trichloroacetic acid during the experiment, the cell membranes and other membranes get broken down into pieces and form vesicle. Means lipids form vesicles which are not water soluble. So these membrane fragments in the form of vesicles get separated with acid insoluble pool. Getting my point? If you will look at this diagram, you will understand we are getting lipid in acid insoluble fraction, but the molecular weight of lipids is less than 800 Dalton, and we are getting these lipids in acid insoluble fractions because the characteristic feature of lipids is lipids form vesicles. That's why they are coming under the macromolecules category. But instead they are micromolecule. They should come under acid soluble pool. Because of the formation of this vesicle, these lipids we are getting in acid insoluble pool or retentate or pallid. So now we move forward. A second experiment was conducted to understand what are the inorganic elements are present in the biomolecules. So to understand this, we have taken the living tissue, we have dried it, the tissue is fully burned. After the tissue is fully burned, we know that all carbon compounds are oxidized to gaseous form, for example CO2 and water vapor, and what is remaining? Remaining is ash. Now after chemical analysis, we found that this ash is composed of inorganic elements like calcium, magnesium, potassium, sodium, water, and compounds like phosphates, sulfates, sodium chloride and calcium chloride. It's not like that biomolecules are only composed of organic molecule. Some inorganic elements are also present in these biomolecules, for example calcium, magnesium, potassium, sodium, and some salts of phosphate salts, sulfate salts, sodium chloride, calcium chloride, etc. So these two experiments gives us clear idea of biomolecules. What are biomolecules? What is their biochemistry? So thank you so much for understanding. Thank you.